Hello, everybody. Thank you so much for coming. We're going to start smack bang on time today because we've got oodles to discuss. So before we start, I would like to acknowledge the traditional owners of the land on which we gather here today, the Turrbal and Yugera people. And I pay my respects to their elders, past, present, and emerging, and extend that respect to all Aboriginal and Torres Strait Islander peoples who are here today. So welcome to Chromosomes and Hormones, Sex-Related Factors to Consider for All Neuroscience Research. My name is Erynn Christensen. I'm a postdoc from the Feinstein Institutes for Medical Research at Northwell Health in New York. And I'm very excited to be your host for today. So there are two main goals of this roundtable. That's to inspire and to inform. We want to inspire you to consider sex and sex hormones and reproductive health in your research. And we want to inform you on how you can meaningfully measure, report, and analyze sex and reproductive health data. So by thoughtfully collecting the right data, we can assist the wider community of science to make inroads into understanding the human brain, not just the male brain and the female brain, but the human brain, and how this relates to disease, risk, and treatments. So we have a fantastic stellar cast of presenters today. We're going to start off with Dr. Erynn Rasnahan, who unfortunately can't be here, but he's made an excellent presentation that we'll show you. So he's going to set the scene and explain why we should be researching sex differences and be considering these things in our brain research. Then we've got Dr. Anne-Kristen Kimmink, and she's going to present the key considerations for hormonal staging in females and males. That will be followed by Dr. Caitlin Taylor and Dr. Winnie Orchard, who are going to talk about tools that we can use to look at hormone, brain, and reproductive health relationships. And finally, I'm going to present some tips and tricks on how we can analyze this data and appropriately report this data in our manuscripts. We'll then open up to a discussion in which we want to hear from you. We want to really have a good back and forth, asking questions to the panel about their experiences working with this sort of data and also hearing about your experiences. So we're all learning as a community, and that's what we want to kind of foster today. So without further ado, we'll start with Professor Erynn Rasnahan.
Hello, I'm Erynn Rasnahan from the National Institutes of Mental Health Intramural Research Program in the US. A huge thanks to the organisers for giving us the opportunity to convene this roundtable and hopefully activate debate and discussion around this important topic. Apologies to you all that I'm not able to be here in person as I'd intended. In this pre-recorded version of my talk, I'm going to try and give a big picture framing of this topic, outlining some of the motivations for looking at sex-based influences on the human brain and some of the important conceptual distinctions one has to hold in mind whilst doing such work. One of the key motivators for looking at sex-based influences on the human brain comes from the field of medicine. Diverse brain-related health outcomes show profound sex differences in their prevalence, presentation, and prognosis. I'm showing several such conditions here ranked by sex differences in prevalence. Now, these differences likely reflect a complex mix of biological and social contributions. But evidence for likely contributions from biological factors, at least in part, comes from the fact that these sex differences occur in a cross-diagnostic manner. They emerge with stereotyped developmental timing, and they're relatively invariant across different historical epochs and societal settings. And this raises the question of whether there may be an intersection between sex-based influences on human brain organization and neuropsychiatric risk. In tackling this question, it's important to use both between and within group lenses. So in the between group lens, it's become clear, for example, in recent work that humans show reproducible sex differences in regional grey matter volume after controlling for sex differences in overall brain size. These brain maps show coloured areas of recurrently reported sex differences in regional grey matter volume across 3 different studies, representing a total of about 5,000 individuals from the UK, US and Germany. Regions in blue tend to be relatively larger in females, and regions in red tend to be relatively larger in males. Now, we don't know if these regional sex differences in grey matter volume have any functional relevance at all, or we don't know what causes them either, but they do seem to be spatially patterned and reproducible, and strikingly concentrated in brain systems that have actually been implicated in risk for diverse sex-biased neuropsychiatric conditions. But it's important to recognise that sex may also modulate health outcomes by creating group-specific sources of variance that needn't necessarily manifest as group differences in brain or features. And this urges a within-group lens. For example, there's growing evidence that female-specific physiological processes can influence brain organization. This is an image depicting changes in functional connectivity linked to estradiol levels over an individual menstrual cycle, but diverse female-specific processes and exposures like usage of oral contraceptive pills, pregnancy and menopause have also been shown to influence brain organization. It's important to consider the ample opportunity too for male-specific sources of hormonal influences on the brain. This plot line at the top of this graph shows the fluctuation in circulating testosterone levels over a day in young males, a high dynamism that is attenuated with age and has a capacity to influence brain-related traits. Finally, sex chromosome dosage, a fundamental biological difference between XX females and XY males, is also known to influence brain organization. The coloured regions in this map show parts of the cortical sheet where surface area is sensitive to how many sex chromosomes you carry, as determined by in vivo neuroimaging analysis across a large group of people with varying numbers of sex chromosomes, including the typical XX and XY dosage, as well as sex chromosome aneuploidies like XXY, XYY, etc. So taken together, the sorts of observations illustrated on this slide underline the capacity of sex and related hormonal and genetic influences to shape many of the variables we care about in neuroscience and in this neuroimaging community. And we're delighted that this roundtable is hopefully providing a space for us all to explore this as a community. As we do explore things though, in a final thought, it'll be important to hold in mind that although hormonal, genetic and external environmental factors can all present sex-based influences on the human brain, there is likely to be an astoundingly complex interaction between all of these processes over developmental time that in humans creates a complex entanglement of sex and gender in a manner that is fundamentally difficult to causally dissect. Well, thanks for your attention and hope you all enjoy the session. Bye.